Hey guys, tonight we are doing something a little different. Tonight we are not going to medieval Europe. We are not going to ancient Peru. We are not going to any battlefield, dungeon, or suspiciously fragrant peasant hut. Tonight we're going somewhere far stranger. We're going inside a small white pill that approximately 1 in 8 Americans has either taken, been offered, or found inexplicably in the back of someone's medicine cabinet during a house party in their 30s. Tonight we are talking about Wellbutrin and bath salts, and the fact that these two things, one a respectable FDA, approved antidepressant prescribed by calm doctors in quiet offices, and the other a substance associated with some of the most unhinged news headlines of the early 2000s, are, from a chemistry standpoint, closer to each other than either of them would probably like to admit. So before you get comfortable, take a second to like the video and subscribe, but only if you genuinely enjoy what we're doing here. No pressure, no guilt, no notification bell, guilt tripping you at 7 in the morning. Just a quiet thanks if this is your kind of thing. Drop a comment telling me where you're listening from and what time it is. If it's somewhere between your second glass of water and your 4th attempt at falling asleep, you're exactly the audience this was made for. And before we begin, let's just sit with a few questions. Have you ever wondered how a drug gets approved? Not the polished version they tell you on the commercial, the version where everyone is running through a meadow in soft focus while a voice calmly lists 37 side effects. The real version. The version involving decades of failed experiments, accidental discoveries, pharmaceutical companies making educated guesses, and the FDA doing its level best to keep up with all of it. Have you ever wondered why Wellbutrin of all antidepressants is the one that helps people quit smoking? Why it's the one that tends not to cause the side effects that make people stop taking antidepressants? Why it has a reputation among people who've taken it for being a little more, how to put this, electrically interesting than the others? Have you ever wondered what bath salts actually are? Not the Epsom salt situation in your bathroom. The other ones. The ones that made headlines in 2012 with a level of collective panic that suggested the entire country had briefly forgotten how chemistry works. Did you know there is a class of molecules, a whole family of compounds, that sits at the intersection of all of these questions? That Wellbutrin and the stimulants in bath salts and cathinone, which is a naturally occurring substance found in a plant that has been chewed in East Africa for centuries, are all chemical cousins, that they share a skeleton, a molecular backbone, the way you and your most chaotic relative share a jawline. Did you know that the story of how we figured all this out involves a Scottish physician, a Cold War-era pharmaceutical company, a plant called khat, a researcher who spent decades quietly cataloguing the effects of compounds that made regulators deeply uncomfortable, and yesment in the early 2000s when synthetic chemistry briefly outran the law? Of course you didn't. You were busy living your life. That's fine. That's what we're here for. Congratulations. You've just enrolled in the most relaxing pharmacology lecture ever recorded. The year is now. The subject is chemistry. The vibe is somewhere between a documentary and a lullaby. And by the time we're done, you will either be asleep or genuinely fascinated, and honestly, both outcomes are completely acceptable. Most people who take Wellbutrin know roughly 3 things about it: that it's an antidepressant, that it's also used to help people quit smoking, and that it works differently from the other ones. Beyond that, the details get hazy fast, and that's not a criticism. Most people don't need to know the molecular mechanics of a drug to take it responsibly, in the same way that most people don't need to understand internal combustion to drive to the grocery store. You put the key in, you go. The car does its thing, the antidepressant does its thing. Life continues. But here's the thing about Wellbutrin specifically. The story of what it actually is, where it came from, and why it does what it does is so unexpectedly strange. so full of wrong turns and accidental discoveries and pharmaceutical plot twists that it deserves to be told slowly at night by someone who finds it as genuinely weird as it is. Let's start at the beginning. Wellbutrin is a brand name. The actual drug is called Bupropion, which sounds, if you say it out loud right now, like a word someone invented while sneezing into a chemistry textbook. Bupropion. It's not glamorous. It doesn't have the sleek, vaguely European confidence of Prozac or the quietly authoritative hum of Zoloft. It sounds like the name of a minor Roman emperor who didn't last long, and yet Bupropion is one of the most widely prescribed psychiatric medications in the world, taken by tens of millions of people across multiple continents for two entirely separate clinical purposes, under two entirely different brand names, with a history so complicated that the label on the bottle barely hints at it. Bupropion is an antidepressant, yes, but it belongs to a class called norepinephrine dopamine reuptake inhibitors, NDRIs, which is a category so sparsely populated that Bupropion is, for most practical purposes, the only member most physicians ever prescribe. Every other major antidepressant on the market targets serotonin, or serotonin and norepinephrine together, or older mechanisms involving enzymes called monoamine oxidases. Bupropion is the one that does the dopamine thing, and that difference is not cosmetic. It is not a minor variation, like choosing the slightly different trim package on the same car. It is a pharmacologically meaningful distinction that produces a clinically different experience for many of the people who take it. Dopamine is the neurotransmitter most closely associated with motivation, anticipation, and reward. Think of it as the brain's internal hype man, the system responsible for making goals feel worth pursuing, for generating the forward pull toward things that matter, for producing the specific feeling that getting out of bed and doing something might actually lead somewhere good. When your brain's dopamine systems are working well, you want things, you look forward to things, you feel the quiet pull of the future. When they're not working well, motivation goes first, then interest, then the capacity to start things, to sustain effort, to feel that a goal is worth the energy it costs. Depression, in its most energic presentations, looks and feels like a dopamine problem, not just a serotonin problem, and Bupropion is the drug in the antidepressant class that specifically addresses that dimension. The smoking cessation effect comes from the same mechanism approached from a different angle. Nicotine is addictive primarily because it floods the dopamine system, fast, reliably, every single time. Bupropion's gentler engagement with the dopamine reuptake system appears to cushion the withdrawal experience, to reduce the amplitude of the craving without replicating the high. It doesn't replace the cigarette. It just makes the absence of the cigarette slightly less unbearable, which turns out to be enough for a meaningful proportion of people who are trying to stop. None of this was discovered on purpose. The story of how Bupropion became a medication is considerably messier, considerably more interesting, and considerably more instructive about how pharmaceutical development actually works than the clean version on the packaging suggests. It begins not with Bupropion at all, but with a tuberculosis ward, patients who started dancing, and a chain of accidental observations that eventually led to a molecule that nobody quite knew what to do with for the better part of 2 decades. The pill in your cabinet has a backstory, and it is genuinely worth knowing. Before it was Wellbutrin, it was Bupropion. Before it was Bupropion, it was compound number 341 in a series of molecules a pharmaceutical company called Burroughs Wellcome was quietly working through in the 1960s and 70s, trying to find something new. The naming of pharmaceutical compounds is a process that combines systematic chemistry, regulatory requirements, and a certain amount of creative optimism that the name might eventually sound like something other than a sneeze. The generic name, Bupropion, is derived from its chemical structure. It contains a butyl group, a propiophenone backbone, and an amino group. Say it fast enough and it almost sounds like a mood. Say it slowly and it sounds like you're describing what happened when a chemistry student knocked over 3 different beakers at once. Either way, it is the name of a compound that has been taken by an estimated 30 million people and that sits, quietly, at the intersection of antidepressant pharmacology, smoking cessation medicine, and one of the stranger episodes in the recent history of drug regulation. Burroughs Wellcome was, by the standards of mid-20th century pharmaceutical companies, an unusual institution. It was established in 1880 in London by 2 American pharmacists, Silas Burroughs and Henry Wellcome, who had the specific, and at the time, moderately radical idea of producing medicines in standardized, compressed tablet form, which they trademarked under the name tabloid. That word, tabloid, is now used almost exclusively to describe a format of newspaper associated with celebrity gossip and alarmist headlines, which is either a tragic brand dilution or an accidentally perfect metaphor for what pharmaceutical marketing occasionally does to pharmacological nuance, depending on how charitable you're feeling. The company that Henry Wellcome built after his partner's early death was research-focused in a way that was unusual for the industry. Wellcome himself was broadly curious about medicine and science, in ways that went beyond commercial application. He founded institutions, collected artifacts, and invested in research that had no clear short-term return. The research culture at Burroughs Wellcome reflected this. Its laboratories were staffed by serious scientists doing serious work, and the company's output included contributions to antiviral medicine, cancer treatment, and gout pharmacology that went well beyond the average pharmaceutical company's ambitions. 2 of its scientists, George Hitchings and Gertrude Elion, won the Nobel Prize in Physiology or Medicine in 1988 for developing a rational approach to drug design based on understanding the biochemical differences between human and pathogen cells. This is the kind of scientific culture in which a medicinal chemist could work on an aminoketone compound for years without being told to drop it and make something more immediately profitable. That patience was not incidental to Bupropion's existence. Nariman Mehta synthesized the compound in 1969 as part of a broader exploration of the aminoketone chemical family. It showed interesting preliminary activity. It affected monoamine reuptake in ways that seemed potentially clinically relevant, but it was not a dramatic winner. It did not produce the kind of results in early animal models that make pharmaceutical executives sit up and reorder priorities. It was interesting. At Burroughs Wellcome, interesting was enough to keep working. The specific target was a cleaner antidepressant, something that engaged the monoamine system without the side effect baggage of the tricyclics or the dietary restrictions of the MAO inhibitors. Bupropion, on paper, offered both. Its receptor profile was different, more selective, less pharmacologically promiscuous, and its mechanism involved dopamine and norepinephrine rather than serotonin, which distinguished it from the emerging class of SSRIs being developed elsewhere. It was, structurally and mechanistically, its own thing, a member of a chemical family that had not previously produced an approved psychiatric medication, sitting in the aminoketone corner of the pharmacological landscape and waiting for someone to figure out what to do with it. What nobody knew, in 1969, was that this compound's structural family would eventually become one of the more discussed topics in drug regulation. That the cathinone scaffold it sat on would, decades later, generate emergency legislative responses on multiple continents. That the same molecular architecture that Mehta had arranged carefully in a Burroughs Wellcome laboratory would later be rearranged by other chemists in other contexts and end up in products sold in head shops labelled as plant food. None of that was visible from the 1969 vantage point, What was visible was a molecule with an interesting profile, a patient company willing to follow it, and a long complicated road between the synthesis and the prescription bottle. To understand why bupropion was considered remarkable, you first need to understand what came before it. And the story of the first antidepressants is one of the great accidental discoveries in 20th century medicine, a story that does not begin in a laboratory with a clear hypothesis and a controlled experiment, but in a tuberculosis ward in the early 1950s, with a drug that was supposed to do something completely different and patients who started dancing. The drug was iproniazid. It had been developed as a treatment for tuberculosis, which in the pre-antibiotic era remained one of the most significant infectious disease burdens in the developed world. Tuberculosis wards were not, generally speaking, cheerful places. They were places of long illness, uncertain prognosis, and a great deal of quiet waiting punctuated by events nobody wanted. In 1952, physicians treating tuberculosis patients with iproniazid began noticing something that absolutely did not fit the expected clinical picture. The patients were getting happier, not just better in the lungs, happier, more energetic, more engaged. Some reports describe patients getting up and dancing in the wards, which was, to put it in the most clinical terms available, not a documented side effect of tuberculosis treatment, and not a behaviour tuberculosis wards were architecturally prepared to accommodate. The observation was reported. It was received with a mixture of interest and scepticism that unexpected clinical observations reliably produce, the same mixture that greets any finding that does not fit the existing theoretical framework, because the existing theoretical framework has a strong prior commitment to its own correctness and does not yield without evidence, the evidence accumulated. Subsequent research identified the mechanism. Iproniazid was inhibiting an enzyme called monoamine oxidase, the enzyme the brain uses to degrade neurotransmitters after they've done their work. By blocking this enzyme, iproniazid allowed neurotransmitters to accumulate, to remain active longer, to produce effects that included, rather dramatically, a lifting of mood in patients who had not been expected to experience anything of the kind. This was the first monoamine oxidase inhibitor, the founding member of a drug class that would eventually include a family of compounds, several of which are still prescribed today. The discovery was significant not only as a clinical tool but as a conceptual opening. It provided the first real biological evidence that depression was a condition with a neurochemical substrate that could be pharmacologically addressed. This was not, in 1952, the consensus view. The consensus view was considerably more psychodynamic, considerably more interested in childhood experiences and repressed conflicts, and considerably less interested in the idea that a pill could address what most of the field considered a psychological problem. Iproniazid changed that conversation, slowly and against considerable resistance. Around the same time, a drug called imipramine was being tested for schizophrenia, a condition it turned out not to treat at all, and was noticed to produce significant mood elevation in patients who also had depression. Imipramine became the first tricyclic antidepressant, named for the three-ring chemical structure at its molecular core, and it became the first widely prescribed antidepressant medication in the world. Two separate accidental discoveries of antidepressant activity through two entirely different mechanisms in the space of a few years. The 1950s were apparently a good decade for not finding what you were looking for and finding something considerably more interesting instead. The drugs that came from these discoveries were genuine clinical advances. They helped people. The effect sizes in patients with moderate to severe depression were real and meaningful. But they were not easy drugs to take, and the profile of difficulty they carried, the side effects of the tricyclics, the dietary restrictions of the MAO inhibitors, shaped the entire subsequent history of antidepressant development, including the question that Burroughs Wellcome's laboratories were eventually trying to answer. Can we do this better? Can we find something that works without costing this much? Bupropion was one attempt at that answer. It was not the last. The accidental discoveries of the 1950s needed an explanation, and what they got, after considerable scientific debate, several competing hypotheses, and the kind of slow consensus building that characterizes fields working at the edge of what they actually understand, was the monoamine hypothesis of depression. It became the dominant theoretical framework in psychiatric pharmacology for the latter half of the 20th century. It is also, in its popular form, one of the most successfully communicated and most significantly oversimplified ideas in the history of medicine. The core of the hypothesis is this: depression is associated with insufficient activity in the brain's monoamine neurotransmitter systems. The relevant monoamines are three: serotonin, norepinephrine, and dopamine. These are chemical messengers the brain uses to communicate between neurons, small molecules released from one cell that travel across a synapse and bind to receptors on the next cell, producing a signal. When monoamine activity is adequate, the systems that these neurotransmitters regulate, mood, energy, attention, motivation, sleep, appetite, the whole anxious human enterprise of getting through a day, run more or less as intended. When monoamine activity is chronically insufficient, those systems break down in ways that produce the recognizable clinical picture of depression. It is a satisfying hypothesis. It is clear, it is communicable, and it provided the conceptual scaffolding that allowed pharmaceutical companies to describe, in accessible terms, what their drugs were doing and why. The pharmaceutical advertising version, the little animation of a synapse with sad-looking serotonin molecules falling into the void, while a soothing voice explains that your antidepressant helps restore the balance, is a direct descendant of the monoamine hypothesis, polished until the complexity has been entirely removed, and what remains is a story clean enough to accompany images of people doing yoga in soft light. The problem is that the hypothesis, in its simple form, is wrong. Not entirely wrong. The monoamine systems are clearly involved in depression, and the drugs that modulate them clearly help many people. But wrong in its suggestion that depression is simply a shortage of neurotransmitters, in the way that iron deficiency anemia is a shortage of iron. The relationship is much more complicated, much more bidirectional, and much more dependent on structural and functional factors in the brain that the original hypothesis didn't account for. The most obvious problem is the timeline. Antidepressants increase synaptic monoamine levels within hours of the first dose. The clinical improvement they produce takes 2 to 4 weeks to develop. If depression was simply a neurotransmitter shortage, and antidepressants simply restored the missing neurotransmitters, you would expect the effect to be roughly as fast as the pharmacological mechanism, the way insulin lowers blood sugar within hours of administration. The weeks-long delay suggests that something more complex is happening downstream of the initial monoamine effect. Changes in receptor sensitivity, neuroplasticity, gene expression, or circuit-level reorganization that take time to develop, and that are the actual substrate of the therapeutic response. What this means for understanding bupropion is important. The drug's mechanism, dopamine and norepinephrine reuptake inhibition, is real, and the monoamine hypothesis provides the framework within which that mechanism is clinically understood. But the actual reason bupropion helps people with depression is probably not simply it increases dopamine and norepinephrine levels in the synapse. It is more likely that the increase in monoamine availability triggers a cascade of downstream effects, changes in prefrontal cortical function, in the regulation of the HPA stress axis, in neuroplastic processes in the hippocampus that constitute the actual therapeutic mechanism. The monoamine story is the beginning of the explanation, not the whole of it. and that caveat applies equally to every antidepressant in every class, regardless of which neurotransmitter system they primarily engage. The early antidepressants worked. That deserves to be said clearly and without qualification before the side effects are catalogued, because the temptation to lead with the problems can obscure the genuinely important thing. These drugs helped people who had not been helped before, at a time when the alternative was primarily institutional care and the quiet persistence of suffering. The progress was real. For many patients, the tricyclics and MAO inhibitors were transformative in ways that justified all the inconvenience and discomfort that came alongside them. But the side effects were considerable, and they were not trivial inconveniences. They were daily, chronic, present from the first dose, and in some cases severe enough to tip the cost-benefit calculation away from treatment before the treatment had fully worked. And understanding them is essential to understanding why Bupropion, with its different mechanism and different side effect profile, represented something genuinely useful rather than just pharmacological novelty for its own sake. The tricyclic antidepressants, the class that includes imipramine, amitriptyline, nortriptyline, clomipramine, and a number of others, produced their therapeutic effects through monoamine reuptake inhibition. But they were pharmacologically promiscuous, which is the polite way of saying that they did not stay in their lane. Along with blocking the monoamine reuptake transporters that produce the antidepressant effect, they also blocked 3 other receptor systems with predictable and unwanted consequences. Blocking muscarinic acetylcholine receptors produced what are called anticholinergic effects: dry mouth, blurred vision, urinary retention, constipation, and, at higher doses or in older patients, cognitive effects including confusion and memory impairment. These were present in essentially all patients at therapeutic doses. Dry mouth, to give just one example, is not a trivial inconvenience when it is present every day, all day, for the duration of treatment, which for many patients meant years. It affects speech, it affects eating, it increases dental decay risk, and it is a constant reminder that you are on medication and that the medication is doing things to your body beyond its primary intention. Blocking histamine. H1 receptors produce sedation and weight gain. The sedation was sometimes therapeutically useful for patients with insomnia as part of their depression, but it was also present during the day in a way that impaired function. The weight gain was consistent and for many patients clinically significant. Not the modest gain of a few pounds that various medications produce, but substantial changes in body weight that introduced their own health implications and that, for patients with self-image concerns already entangled with their depression, added another layer of difficulty to the experience of treatment. Blocking alpha-adrenergic receptors caused orthostatic hypertension, a drop in blood pressure when moving from sitting or lying to standing that produced dizziness and, in older patients, created an increased fall risk that was serious enough to limit prescribing in that population. And the cardiac conduction effects of the tricyclics, their ability to slow electrical conduction through the heart, required monitoring in patients with pre-existing cardiac conditions, and made these drugs genuinely dangerous in overdose, which was a particular concern in a patient population with elevated suicide risk. The MAO inhibitors had a different problem set, centered almost entirely on the dietary restriction. Because monoamine oxidase in the gut metabolizes tyramine, found in aged, fermented and cured foods, blocking that enzyme with an irreversible MAO inhibitor meant that dietary tyramine entered the bloodstream intact, where it could cause a sudden, severe hypertensive crisis. The list of foods to avoid was extensive and socially significant. Aged cheeses, cured and fermented meats, certain wines and beers, soy products, broad beans, and various others. Patients were given cards to carry. They modified their diet substantially. And they did it indefinitely because the irreversible MAO inhibitors continued to block the enzyme for weeks after the last dose until new enzyme was synthesized. This was, to put it plainly, a lot to ask of people who were already struggling. The drugs worked, and they asked a great deal in return. Burroughs, Wellcome, the company whose name lives on, slightly rearranged in the brand name Wellbutrin, was, by the standards of mid-20th century pharmaceutical companies, genuinely unusual. Not unusual in the sense of eccentric or difficult to work with, but unusual in the deeper sense of being organised around values that were not exclusively commercial. Research for its own sake, scientific rigour ahead of short-term return, and an institutional culture that took the long view in a field that frequently rewards impatience. These qualities were not incidental to the development of Bupropion. They were the reason Bupropion survived long enough to become a medication. The company had been established in 1880 in London by Silas Burroughs and Henry Wellcome, two Americans who had arrived in Britain with a specific commercial idea, producing medicines in standardized, compressed, measured tablet form, and the entrepreneurial confidence to execute it in a market that had not previously demanded it. Their trademarked name for these compressed tablets was tabloid, a word that has since migrated so thoroughly into another context that its pharmaceutical origins require explanation. The migration of a pharmaceutical brand name into the vocabulary of sensationalist journalism is either a coincidence or the universe's way of making a point about what happens to clean ideas when they encounter the pressures of mass distribution. Henry Wellcome outlived his partner by decades and built the company into something considerably larger than its original commercial premise. He was, by all accounts, a man of broad intellectual curiosity who collected historical medical artifacts, founded institutions, and invested in research that had no clear short-term application. The Wellcome Library and the Wellcome Collection in London, both still operating and still significant, are monuments to this curiosity. The research culture he established at the company reflected the same values: patient, rigorous, willing to follow a scientific thread into territory that hadn't been commercially mapped. This culture produced results. The Burroughs Wellcome laboratories in the 20th century contributed significantly to antiviral pharmacology, to the treatment of gout, to cancer chemotherapy, and to the development of immunosuppressive drugs. George Hitchings and Gertrude Elion, who worked at the company for decades and won the Nobel Prize in Physiology or Medicine in 1988, did so for developing a rational approach to drug design. The idea that you could target drugs to specific biochemical pathways by understanding the differences between human and pathogen biochemistry, rather than by screening compounds randomly and hoping for useful results. This was, for its time, a conceptually transformative approach. It is now standard practice. Into this environment came the aminoketone program of the 1960s and 70s, the research effort that produced Bupropion. The program was not the company's most prominent work. It was not generating Nobel-level results in the early years. It was a systematic exploration of a chemical family that had pharmacologically interesting properties, and that represented one possible answer to a clinical question that the field was actively asking: is there a better antidepressant? Not better in the sense of dramatically more effective. The tricyclics and MAO inhibitors were effective enough, but better in the sense of safer, cleaner, more tolerable over the long-term treatment courses that depressive illness frequently requires. The aminoketone family, cathinone-related compounds, substituted phenethylamines with a ketone at the beta-carbon, was a reasonable place to look for that answer. The family had documented monoamine activity. The structure-activity relationships were not fully mapped, which meant there was meaningful work to do in systematically exploring which modifications produced which pharmacological profiles. And Burroughs Wellcome had the resources, the patience and the scientific culture to do that systematic work without demanding results on a timeline that the chemistry wasn't ready to meet. Nariman Mehta's synthesis of Bupropion in 1969 was not a dramatic moment. It was one data point in a larger exploration, but it was the right data point in the right company at a moment when the clinical need it addressed was real and the institutional willingness to follow it through was present. Those conditions do not always align. When they do, occasionally, a compound that starts as item number 341 in a research series ends up in 30 million medicine cabinets. The synthesis of Bupropion in 1969 is attributed to Nariman Mehta, a medicinal chemist working in Burroughs Wellcome's research program on aminoketone compounds. His name appears in the original patent alongside other researchers involved in the broader program, and it is from that patent that the compound's pharmaceutical journey begins, quietly, without fanfare, as one of many compounds being synthesized, characterized and evaluated in a systematic search for clinical utility. Medicinal chemistry is not glamorous work, even when it produces significant results. It is largely a practice of systematic variation, of taking a molecular scaffold with known activity, making methodical changes to its structure, testing each variation, and building a picture of which structural features are responsible for which biological effects. This picture, the structure-activity relationship, is the foundation of rational drug design, and it is constructed compound by compound, one synthesis at a time, in a process that is closer to careful craftsmanship than to inspired discovery. The inspired discoveries do happen, but they tend to be recognized as such only in retrospect. The scaffold Mehta was working with was the cathinone skeleton, a phenethylamine backbone with a ketone group at the beta carbon, the molecular core of the compound found naturally in the khat plant, and the structural parent of the entire cathinone family. This scaffold had documented stimulant and monoamine active properties. It engaged the dopamine and norepinephrine transporter systems in ways that were pharmacologically relevant. The question was whether specific modifications to the scaffold could tune that activity toward clinical utility, toward the monoamine reuptake inhibition that was the therapeutic target for antidepressant effects, while away from the stimulant properties and addiction liability that made the parent compounds clinically problematic. Bupropion represented one specific answer to that question. Its tertiary butyl group on the nitrogen, a large branch carbon structure, was not an arbitrary choice. Bulkier amino substituents generally reduce the affinity of the compound for monoamine transporters and reduce stimulant potency, which was exactly the direction the medicinal chemistry was intended to go. The chlorine substituent on the aromatic ring affected the compound's electronic properties and metabolic profile in ways that were predictable from the structure-activity relationships of the broader phenethylamine family. The specific combination of these modifications produced a compound that was weaker at the dopamine transporter than the parent cathinone, more selective for norepinephrine relative to dopamine, and without the potent direct-releasing activity that characterized the amphetamines. In early testing, this profile looked interesting. The compound affected monoamine reuptake in ways that suggested antidepressant potential. It did not have strong activity at the receptor systems responsible for the tricyclic side effects. It was not a potent direct dopamine releaser. It was not as stimulant in character as its cathinone relatives. It was, in the pharmacological vocabulary of the time, cleaner. Not without activity, but more targeted in the activity it had. What it was not, in 1969, was a finished drug. It was a promising compound in a preclinical program, a candidate, not a medication. The distance between those two things is where the vast majority of drug candidates disappear, undone by toxicity discovered in animal studies, by lack of efficacy in human trials, by manufacturing problems, by regulatory concerns, by the accumulated weight of the gap between what looks good in a test tube and what works safely in a human being with a complicated biology and a life to return to. Bupropion crossed that distance. It did so slowly, with significant setbacks, and in ways nobody fully predicted. But it crossed it, because the initial profile was good enough to justify continued investment. The company behind it had the patience to sustain that investment through the difficult periods, and the clinical need it addressed never went away. Nariman Mehta synthesized a molecule. What it became, the medication in the amber bottle, the smoking cessation tool, the structural relative of both khat and bath salts, the compound at the centre of one of the stranger stories in modern pharmacology, was the work of decades and dozens of people who followed the chemistry wherever it led. Here is where we need to look at the molecule itself, because the shape of bupropion, the actual physical arrangement of its atoms, is the key to understanding everything that follows. The bath, salt's connection, the smoking cessation effect, the specific quality of its clinical impact, the reason it occupies the unusual position it does in both pharmaceutical prescribing and the history of recreational drug regulation. All of it flows from the structure, and the structure belongs to a family. Bupropion is a substituted cathinone. This is a classification, not a criticism. Cathinone is the naturally occurring stimulant compound found in the khat plant, a compound that the United States Drug Enforcement Administration placed on Schedule 1 in 1993, meaning it is classified as having no accepted medical use and high potential for abuse. Bupropion is a Schedule 5 medication, meaning it is an approved pharmaceutical with accepted medical uses and relatively low abuse potential. Both designations apply to compounds built on the same molecular scaffold. That is not a paradox. It is a demonstration of how much structural modification can change a compound's pharmacological and clinical character, and how human regulatory categories interact with chemistry in ways that are always approximate. The cathinone scaffold at the core of both compounds is a beta-ketophenethylamine. Start with phenethylamine, a benzene ring attached to a 2-carbon chain ending in an amino group. Add a ketone at the beta carbon, the carbon directly adjacent to the amino group, and you have the cathinone skeleton. It is not a complex structure. It is a relatively small, relatively simple molecule, and its pharmacological power derives not from structural complexity, but from structural complementarity. It fits, with varying degrees of precision depending on the specific substitutions, into the binding sites of the monoamine transporter proteins that regulate dopamine, norepinephrine, and serotonin activity in the brain. Bupropion adds 2 specific modifications to this core: a tertiary butyl group on the nitrogen, replacing the hydrogen atoms that sit on cathinone's nitrogen with a bulky branched 4-carbon structure, and a chlorine atom on the aromatic ring at the para position. These are not arbitrary decorations. The tertiary butyl group, by virtue of its size, reduces the compound's affinity for the dopamine transporter. This is a deliberate pharmacological strategy. You want some dopamine transporter activity, because that is part of the therapeutic mechanism, but you want it to be modest and sustained, rather than intense and acute. The bulky amino group achieves the former while reducing the likelihood of the latter. The chlorine substituent affects the molecule's lipophilicity, its tendency to dissolve in fat rather than water, which influences how readily it crosses the blood-brain barrier and how it distributes in body tissue. It also affects the molecule's metabolic profile, influencing which enzymes process it and at what rate. Chlorine on an aromatic ring is a standard medicinal chemistry tool for tuning pharmacokinetic properties, and its presence in bupropion structure reflects the same systematic thinking about optimization that the tertiary butyl group reflects. The result of these modifications measured against the parent cathinone structure is a compound with substantially reduced dopamine transporter affinity, relatively higher norepinephrine transporter affinity, and a pharmacokinetic profile suited to chronic oral dosing. It is Bupropion's specific combination of these properties, not any single structural feature, that produces its clinical character. Take away the tertiary butyl group, and you have a more potent, more stimulant compound with higher addiction liability. Take away the chlorine, and you change the metabolic profile in ways that alter the drug's duration of action and the concentrations of its active metabolites. Every atom in the structure is doing something. The structure is the story. The phenethylamine backbone that sits at the core of Bupropion, cathinone, and the synthetic cathinones is not a chemically obscure structure found in the specialty sections of academic journals. It is one of the most consequential molecular arrangements in all of neuropharmacology, a simple structure that turns out to be extraordinarily good at interacting with the brain's regulatory systems. And that sits at the centre of a family of compounds so pharmacologically significant and so culturally diverse that tracing its members is like drawing a map of everything humans have ever found interesting about consciousness. Start with the backbone itself, a benzene ring attached to a 2-carbon chain ending in an amino group. That's the whole thing, a ring, a chain, a nitrogen. Phenethylamine in its unmodified form occurs naturally in the human brain. As a neurotransmitter and neuromodulator, your brain makes it, uses it, and degrades it rapidly, which is why its effects are difficult to study directly. It also occurs in chocolate, which is either a deeply satisfying coincidence or the universe's way of explaining why certain people have strong feelings about chocolate that go beyond simple flavour preference. Now start making changes to that backbone. Add a methyl group to the alpha carbon, the carbon directly attached to the amino group, and you have amphetamine. A single methyl group, and you have one of the most studied, most regulated, and most controversial stimulant compounds in the history of pharmacology, used therapeutically for ADHD and narcolepsy, used recreationally for its euphoric and performance-enhancing effects, and carrying a cultural and regulatory history that spans a century. Add another methyl group to the nitrogen, and you have methamphetamine, more potent, faster acting, with a profile of abuse liability that has made it the subject of federal law enforcement priorities and documentary series alike. Add a methylene-deoxy bridge across the 3 and 4 positions of the ring, a specific structural modification that creates a 5-membered ring fused to the benzene ring, along with a methyl group at the alpha carbon, and you have MDMA. Empathogenic, serotonergic as well as dopaminergic, associated with rave culture and now, increasingly, with clinical research into PTSD treatment. 1 bridge, 1 methyl group, and the compound moves from the stimulant class to the entactogen class, with dramatically different subjective effects and a dramatically different regulatory and clinical history. Add the beta-keto group that converts phenethylamine to cathinone, and you're in the structural territory that contains both bupropion and the synthetic cathinones, the family we've been discussing, where a single functional group at the beta-carbon is the defining modification that separates this branch of the phenethylamine tree from the amphetamines. Dopamine itself is a phenethylamine, so is norepinephrine, so is adrenaline. The brain's own signalling molecules are built on this scaffold, which is exactly why exogenous phenethylamines are so pharmacologically active. They are structural relatives of the brain's native chemical language, and the receptor and transporter systems that evolved to handle endogenous phenethylamines recognize the synthetic and botanical variants because the structural resemblance is close enough to generate interaction. The drugs work because the brain cannot fully distinguish them from itself. This is the molecular reason that the phenethylamine family has been at the centre of both therapeutic pharmacology and recreational drug use for over a century. The scaffold interacts with the systems that regulate how we feel, how energized we are, how motivated we are, how much we experience pleasure and anticipation. Modifying it produces compounds with varying degrees of access to those systems, varying degrees of intensity in their effects, and varying profiles of risk and benefit. Bupropion is at the mild, controlled, therapeutically useful end of this family. Its structural relatives span a range that goes considerably further in multiple directions. The family tree is real, the differences within it are real, and both need to be understood to understand why any individual member of it is the thing it is. Before there were synthetic cathinones, before there were bath salts, before there were DEA scheduling decisions and emergency legislative sessions and television segments in which journalists struggled to explain chemistry to audiences who had not asked to think about chemistry, there was a plant, Catha edulis, khat, a shrub, growing in the highlands of East Africa and the Arabian Peninsula, catching the sun at altitude, entirely indifferent to the regulatory frameworks that would eventually be constructed around the compound in its leaves. Khat has been used by people for a very long time. The archaeological and historical evidence suggests continuous use for at least several centuries, and possibly considerably longer, in the communities where it grows and where it has always grown. The practice is straightforward in its mechanics. You take fresh leaves and young stems, you chew them slowly, holding the material in the cheek as it releases its active compounds, and over the course of an hour or two, you absorb cathinone and cathine, a related but less potent compound, through the oral mucosa into the bloodstream and from there into the brain. The effects are stimulant in character: increased energy and alertness, reduced appetite, a sense of mild euphoria, and increased sociability. Enhanced fluency of thought, or at least the subjective experience of it, which is not always the same thing but is socially sufficient to produce extended animated conversation. These effects are real and pharmacologically consistent with cathinone's mechanism. It is a monoamine reuptake inhibitor, structurally similar to amphetamine, and its stimulant effects are the predictable consequence of that mechanism. They are also, in the context of traditional use, relatively mild, not because cathinone is a weak compound in absolute terms, but because the oral route of administration through the whole plant, with its associated fibre and enzyme content, produces a slower and more gradual absorption than isolated compound by other routes. The experience of chewing khat leaves for several hours in the afternoon is pharmacologically quite different from the experience of taking a concentrated synthetic cathinone at a higher dose by a faster route. The social context of traditional khat use is not peripheral to understanding the compound. In Yemen and the Horn of Africa, the afternoon khat session is a structured social event with its own etiquette, its own timing, and its own cultural meaning. Men gather after the midday prayer or meal, in a room set aside for the purpose, and spend several hours chewing, talking, listening, and passing time together. Business is conducted, disputes are aired and sometimes resolved. Stories are told. The khat session functions socially as a combination of the coffee house, the pub, and the town meeting, organised around a mildly psychoactive substance whose use is entirely normalised within the cultural framework that produced it. This context is significant when considering the regulatory treatment of khat in Western countries, because the regulatory treatment was largely conducted without adequate reference to it. Khat was evaluated as a drug, as a substance with a psychoactive compound that produced stimulant effects, and regulated on that basis, without consistent acknowledgement that the compound's effects in the context of traditional use looked considerably different from its effects in the context of isolated compound use, that regulatory frameworks were designed to address. The United States scheduled cathinone in 1993. The United Kingdom kept khat legal until 2014. The resulting patchwork of different regulatory approaches in different jurisdictions reflects not a principled scientific assessment of the evidence, but the contingent political and social pressures of different regulatory moments in different places, and through all of it, Bupropion sat in pharmacies, prescribed by physicians covered by insurance, entirely separate from the regulatory conversation about cathinone, because its pharmaceutical status placed it outside the scheduling framework that applied to the natural compound and the synthetic derivatives. Same scaffold, different world. The plant that started something started it long before the pharmaceutical industry arrived, and the regulatory responses it eventually generated had more to do with geography and politics than with the chemistry that connects it, in the most direct structural terms, to an antidepressant that several million people took this morning without giving the connection a moment's thought. Khat has been used in East Africa and Yemen for so long that its history as a cultural practice predates most of the nation-states currently debating whether to prohibit it. This longevity is worth pausing on, because the framing of khat as a drug problem is a relatively recent construction, and understanding how that framing developed tells us something important about the intersection of pharmacology, culture, and regulatory politics, and about how the same compound can be simultaneously a traditional social practice, a public health concern, a scheduling priority, and a structural relative of an FDA-approved antidepressant, depending entirely on which angle you approach it from and which country you happen to be standing in. In the communities where khat use has been traditional, it has not historically been classified as drug use in the Western, regulatory sense of that phrase. It has been classified as a cultural practice, something people do, socially, in structured contexts, with established norms about appropriate use and clear social sanctions against inappropriate use. The khat session in Yemeni or Somali culture has the social function that coffee has in Italian culture, or that alcohol has in Irish culture. A substance-centered gathering practice with its own rituals, its own etiquette, and its own deeply embedded role in community life. The substance is mildly psychoactive. The practice is highly social. The norms governing both are the product of centuries of cultural negotiation, not the product of regulatory intervention. When significant diaspora populations from East Africa and Yemen established themselves in Europe, particularly in the United Kingdom, where there were large Somali, Ethiopian and Yemeni communities by the 1980s and 90s, they brought khat with them. Fresh khat was flown in weekly from East Africa, sold in specialist shops in areas of diaspora concentration, and used primarily within those communities in ways that broadly reproduced the cultural context of origin. It was not a substance marketed to the general population. It was not appearing in mainstream drug markets. It was, for all practical purposes, a community-specific cultural practice that happened to involve a compound with stimulant properties. The regulatory responses varied so dramatically across different Western jurisdictions that they constitute a natural experiment in how political and social context shapes drug policy independent of pharmacological evidence. Sweden classified khat as a controlled substance in 1989. Norway followed. The Netherlands, despite a generally liberal drug policy approach, banned it. The United States scheduled cathinone, the active compound, in 1993, which technically made khat illegal as a source of cathinone, though enforcement was inconsistent, and the legal status of the plant material itself remained genuinely complicated by the co-presence of cathine, a less potent and unscheduled related compound. The United Kingdom maintained khat as a legal substance until 2014, a position that was actively supported for many years by public health researchers who argued, with evidence, that criminalisation would be more harmful to the communities involved than the drug use itself. The research base did not support classification as a high-harm substance. The Advisory Council on the Misuse of Drugs, the UK's official scientific advisory body on drug policy, recommended against classification in 2005 and again in 2013. The government classified it anyway in 2014, citing concerns about use by young people and the risk of the UK becoming a hub for khat trafficking to countries where it was already illegal. The Advisory Council's chair described the decision as based on political rather than scientific grounds. What khat's regulatory history illustrates, sitting alongside the Bupropion story, is that the chemistry of cathinone does not determine its regulatory fate. The regulatory fate is determined by the political and social context surrounding the chemistry, who is using it, where they are using it, how it interacts with existing cultural anxieties, and whether the populations most affected by the regulation have the political influence to shape the policy. Bupropion moved through the FDA process because it was developed by a major pharmaceutical company, studied in clinical trials, and presented within the regulatory framework that the FDA was designed to evaluate. Khat moved through a different set of processes entirely, ones shaped by immigration politics, cultural unfamiliarity, and the intersection of racial anxieties with drug policy that has characterized Western drug regulation throughout its history. Same molecular family, radically different journeys. The chemistry does not explain the difference. Everything else does. In the early 1980s, Bupropion was moving through the FDA approval process with the cautious optimism that pharmaceutical companies develop after years of watching promising compounds fail at the final hurdle, an optimism that is not naive exactly, more like the specific hope of someone who has checked the weather forecast 3 times and is still not entirely sure they won't get rained on. The clinical trials had shown Bupropion to be effective. The side effect profile was different in the ways that mattered. The case for approval was, on the available evidence, solid, and then everything got significantly more complicated. The approval process itself had gone as expected. Bupropion had been in clinical development since the early 1970s, a development timeline that reflected both the genuine complexity of establishing antidepressant efficacy in clinical trials and the specific challenges of characterizing a compound with an unusual mechanism in a field that had not yet fully standardized its outcome measures. The trials showed effect sizes comparable to the tricyclics, with the cleaner side effect profile that had been the goal from the beginning. No significant anticholinergic effects, no dietary restrictions, no cardiac conduction concerns at therapeutic doses, no sexual dysfunction in the clinical trial populations, which would turn out to be one of the drug's most clinically significant advantages in the years after the SSRIs established sexual dysfunction as one of the primary reasons patients stopped antidepressant treatment. The FDA approved Bupropion in 1985 under the brand name Wellbutrin. The name was chosen to echo the company name, Burroughs Wellcome, in a way that was sufficiently indirect to function as a brand rather than a direct label. Wellbutrin. It sounds like the name of a small English village where nothing dramatic ever happens, which turned out to be somewhat aspirational as a naming choice, because the next 2 years were about to be extremely dramatic. Post-marketing surveillance is the monitoring system that operates after a drug is approved and prescribed in the real world, a system designed to detect safety signals that clinical trials, with their carefully selected patient populations and controlled conditions, may not have captured. The clinical trial populations for Bupropion had not adequately represented patients with eating disorders, specifically bulimia nervosa, for reasons that reflected standard trial design practices of the time rather than any deliberate exclusion. Patients with bulimia were not sought out. They were not excluded either. They were simply underrepresented in the data that formed the basis of the approval. Post-marketing prescribing, however, does not replicate trial conditions. Physicians prescribed Wellbutrin to the full range of patients presenting with depression, including patients with eating disorders, and in that population seizure reports began accumulating. The rate was higher than in the trial populations. The distribution of cases had a pattern that pointed toward a specific vulnerability. Patients with bulimia nervosa, who have altered electrolyte balance and metabolic profiles as a consequence of purging behaviours, had a physiologically lower seizure threshold that interacted badly with Bupropion's dose-dependent pro-convulsant properties. In 1986, Burroughs Wellcome voluntarily withdrew Bupropion from the market. This decision, voluntary rather than compelled, undertaken before regulatory action required it, reflected a combination of genuine concern about patient safety and a calibrated assessment of the alternatives. The voluntary withdrawal was accompanied by a commitment to reformulation, to developing extended-release versions of the drug that would deliver the same total dose more slowly, reducing the peak plasma concentrations responsible for the seizure risk, while maintaining the average concentrations needed for therapeutic effect. The withdrawal was a pause, not an ending, but it was a pause that cost 3 years, significant resources, and whatever commercial momentum the drug had been building since its initial approval. By the time Bupropion returned to market in 1989, the pharmaceutical world had acquired Prozac. The rules of the game had changed. The seizure risk associated with Bupropion is real, it is dose-dependent, and it is, once properly characterized and managed through appropriate prescribing and formulation, clinically manageable. But the process of getting from the initial safety signal to the managed risk involved years of research, a reformulation effort that required developing new drug delivery technology, and a period during which the drug's entire future was genuinely uncertain. And understanding this period is important, because it shaped everything that came after, the specific formulations available today, the prescribing guidelines that govern their use, and the clinical profile that distinguishes Wellbutrin from the other antidepressants that were available when it came back to market. The seizure risk became apparent through post-marketing surveillance after the initial approval in 1985. The signal was a higher than expected rate of seizure reports, and the distribution of those reports pointed towards specific patient characteristics, higher doses, and the specific physiological vulnerability of patients with eating disorders. The mechanism underlying the seizure risk involves Bupropion's effects on neuronal ion channels, specifically at high concentrations, an inhibition of channels that maintain the electrical stability of neurons and contribute to the regulation of seizure threshold. This effect is concentration dependent, which means it becomes clinically significant only when plasma levels exceed a certain threshold. This concentration dependence was the key to solving the problem. If the seizure risk was related to peak plasma concentrations, the high point reached when the drug is absorbed quickly after a dose, then a formulation that delivered the same total dose more slowly would flatten the peak without sacrificing the average concentration needed for therapeutic effect. Extended-release formulations work by embedding the drug in a matrix that releases it gradually as the tablet passes through the gastrointestinal tract, replacing the rapid absorption of an immediate-release tablet with a sustained, slower release that produces a flatter pharmacokinetic profile. Developing such a formulation for Bupropion required solving a specific technical challenge. Bupropion is a water-soluble compound, which makes sustained-release formulation more difficult than for lipophilic compounds that can be embedded in hydrophobic matrices. The approach is eventually developed. First, the sustained-release formulation dosed twice daily. Later, the extended-release formulation dosed once daily, required considerable pharmaceutical engineering work and represented genuine advances in drug delivery technology, not just the application of off-the-shelf solutions. The clinical consequence of the reformulation was a substantially reduced seizure risk at therapeutic doses. The incidence of seizures with Wellbutrin XL at the standard starting dose of 150 mg is approximately 0.1%, 1 in 1,000 patients, which is comparable to the seizure risk of other antidepressants and represents a clinically acceptable level of risk within the overall benefit-risk assessment of the medication. The residual risk is highest at doses above 300 mg per day, in patients with conditions that lower the seizure threshold, and in patients taking other medications that interact with Bupropion's metabolism. Prescribing guidelines address all of these considerations with specific recommendations. The 3 years between the voluntary withdrawal and the return to market were not wasted time. They produced a better formulation, a more thoroughly characterized safety profile, and a set of prescribing guidelines that gave physicians the information they needed to use the drug appropriately. They also produced something less tangible but equally important, an institutional record of how a pharmaceutical company can respond to a safety signal in a way that prioritizes patient welfare over commercial continuity. Whether that record reflects genuine corporate ethics or sophisticated risk management, probably both. In proportions that are difficult to disentangle from the outside, it produced an outcome that was better for patients than the alternatives would have been. The drug came back safer. That is what matters most. 1989, when Bupropion returned to the market as Wellbutrin SR, was a specific moment in the history of psychiatric pharmacology, a moment defined primarily by what had happened 2 years earlier. Prozac had been approved in 1987. The SRI era had begun, and the pharmaceutical and clinical landscape that Bupropion was returning to was not the one it had left. The SRIs, selective serotonin reuptake inhibitors, were genuinely better tolerated than the tricyclics, and their arrival represented a real improvement in the practical experience of antidepressant treatment for a large proportion of patients. The side effect reduction was meaningful. The safety in overdose was substantially better. The prescribing simplicity, no dietary restrictions, no cardiac monitoring required, a flat dose response curve that made titration relatively straightforward, made them easier to use in primary care settings, which dramatically expanded the population of patients who received pharmacological treatment for depression. This expansion was a public health benefit. Many people who needed treatment and had not previously received it began receiving it. Whatever the limitations of the SSRI narrative, and there are significant limitations, both pharmacological and cultural, the reduction in suffering that followed the broader availability of tolerable antidepressants was real. Fluoxetine, Prozac, became something beyond a medication. It became a cultural phenomenon. The 1993 book Listening to Prozac by psychiatrist Peter Kramer turned the drug into a lens through which questions about identity, authenticity, and the pharmacological shaping of personality were publicly debated. The phrase cosmetic psychopharmacology entered the vocabulary. The idea that antidepressants might not just treat illness but enhance normal functioning, make people better than well, was both celebrated and contested. Prozac was on the cover of Newsweek. It was discussed on talk shows. It became, for a period, shorthand for a whole new relationship between psychiatry and the general public, one in which the brain's chemistry was discussable. The pharmacological management of mood was normalized, and the stigma of mental health treatment, while far from eliminated, had been meaningfully reduced. Into this landscape came Bupropion, a drug with a different mechanism, an unusual history, and clinical advantages that were real but harder to communicate in the simple language the SSRI narrative had established. It did not have an easy story. Restores serotonin levels is 4 words that most people can follow without difficulty. Inhibits dopamine and norepinephrine reuptake through a mechanism distinct from the amphetamines despite structural similarities to the cathinone family, is a sentence that requires significantly more effort to process, particularly before 9 in the morning. The commercial consequence was that Bupropion established itself in a specific clinical niche, rather than competing head-to-head with the SSRIs for the broad antidepressant market. It became the drug of choice for patients for whom SSRI side effects were intolerable, particularly sexual dysfunction, which emerged as one of the most clinically significant barriers to long-term antidepressant adherence. It became the preferred option for patients with the low-energy, low-motivation presentations of depression, where the dopamine component was specifically relevant. It became the add-on agent for patients who had partial responses to SSRIs, the drug that restored motivation and energy in patients whose mood had improved on an SSRI but whose ability to function had not fully recovered. None of this was a consolation prize. These were real clinical applications serving real patient needs. But they were a different kind of commercial success than Prozac's, quieter, more specialized, less culturally prominent. The drug that returned in 1989 was, pharmacologically, the same compound that had been approved in 1985. Everything around it had changed. It adapted to that change by finding the spaces in the market where its specific profile was most useful, and it built a clinical presence that has lasted decades. Not loudly, just steadily, which, for an antidepressant, is arguably exactly the right approach. The observation that Bupropion helped people quit smoking emerged from clinical practice in the early 1990s, the way many of the most significant findings in pharmacology emerge, not from a designed experiment with a pre-specified hypothesis, but from someone paying attention to something they hadn't been asked to look for, a pattern noticed in passing, a note in the margin of a clinical encounter, the kind of observation that gets made because a physician is curious enough to wonder why. A patient mentioned something unexpected and honest enough to write it down rather than filing it under coincidence and moving on. The observations were accumulating across multiple practices. Physicians prescribing Wellbutrin for depression were hearing, in the course of routine follow-up appointments, that patients were smoking less. Not all of them, not dramatically, not in every case, but with enough consistency across enough patients to constitute a pattern rather than an anecdote. The language in the clinical notes varied, but the theme was consistent. Patients reported that the cigarettes were less appealing, that the urge was quieter, that they had stopped, without particularly intending to, or without the grinding, effortful quality that characterized most quit attempts. One patient's description, "I just sort of forgot to want one," turned out to be remarkably precise pharmacological reporting dressed up as casual conversation. The mechanism that made this plausible was already partially understood. Nicotine's addictive power rests primarily on its ability to produce rapid, reliable dopamine release in the brain's reward circuitry. When nicotine binds to nicotinic acetylcholine receptors in the ventral tegmental area, it triggers a cascade that results in dopamine release in the nucleus accumbens, the brain's primary reward processing region. This dopamine signal is fast, it is strong, and it is consistent. Every cigarette reinforces the association between smoking and reward with a reliability that the brain's learning machinery finds extremely compelling. The craving that develops in nicotine dependence is, at its neurochemical core, the dopamine system demanding repetition of the signal it has learned to expect. Bupropion engages the dopamine reuptake transporter, weakly, gradually, through the slow kinetics of the oral extended-release formulation, in a way that maintains a modest elevation in dopamine availability in the reward circuitry without producing the acute surge that nicotine provides. The hypothesis, as it was eventually formalized, was that this sustained dopamine support reduced the amplitude of the nicotine withdrawal experience by partially filling the dopaminergic gap that nicotine cessation created. The craving was not eliminated. That would require replicating the nicotine effect, which bupropion could not and was not designed to do. But it was reduced in intensity, made more manageable, brought down from a physiological imperative to a preference that could be resisted with ordinary human willpower rather than requiring extraordinary effort sustained indefinitely against a pharmacological tide. The clinical trial that confirmed the smoking cessation effect was published in the New England Journal of Medicine in 1997. It was a randomized controlled trial that compared bupropion, nicotine patch, the combination of both, and placebo across several hundred patients over 12 months. The results were clear enough to be convincing. Bupropion more than doubled the cessation rate compared to placebo at 12 months. The combination of bupropion and nicotine patch performed better than either alone. The effect sizes were large enough to be clinically significant and consistent enough across the trial population to be generalizable. The public health implications were substantial. Smoking cessation is one of the highest impact health interventions available in terms of reduction in all-cause mortality and morbidity. A pharmacological tool that reliably doubled cessation rates, represented an enormous quantity of prevented heart disease, prevented lung cancer, and prevented years of life lost, not in the abstract but in real patients in real practices around the world. It also gave bupropion a second life, a new population, a new indication, and a commercial trajectory that its performance as a standalone antidepressant in the SSRI era had not fully established. The accident of the observation turned out to be one of the more consequential accidents in the history of the drug. In 1997, the FDA approved bupropion for smoking cessation under the brand name Zeban. The molecule was Wellbutrin. The dose was equivalent to the antidepressant dose. The pharmacological mechanism was identical. The side effect profile was the same. The prescribing information contained the same warnings. The only substantive differences between Wellbutrin and Zeban were the name on the packaging, the clinical indication on the prescription, the way the drug was marketed, and the insurance coding that determined how different patients were reimbursed for it. Which is to say, everything that mattered to the pharmaceutical company's commercial strategy, and relatively little that mattered to the molecule's chemistry. The decision to brand bupropion separately for smoking cessation was not arbitrary. It was a calculated response to several clinical and commercial realities that, taken together, made a second brand name the sensible approach, even if it also happened to be the profitable one. The clinical reality was that patients seeking smoking cessation treatment were a different population from patients seeking antidepressant treatment, with different framings of their problem, different expectations of their medication, and different relationships with the stigma that remained attached to psychiatric pharmacology in the late 1990s. A patient who wanted help quitting cigarettes did not necessarily want to be told that their cessation medication was, at its core, an antidepressant. Not because antidepressants are bad, they are not, but because the implied message that the smoking addiction was somehow a manifestation of depression rather than a discrete pharmacological dependency was both clinically inaccurate and experientially alienating for patients who did not identify as depressed. Zeban, positioned as a smoking cessation medication that worked through the brain's chemistry, avoided that implication. It acknowledged the pharmacological dimension without the psychiatric framing. It was a pragmatic decision about communication rather than a substantive difference in the drug. Whether this kind of therapeutic rebranding constitutes helpful communication or pharmaceutical sleight of hand is a question on which reasonable people differ, and both positions have something to recommend them. The commercial reality was straightforward. Different insurance categories, different reimbursement structures, and different prescribing environments for psychiatric and smoking cessation medications meant that a single brand covering both indications would navigate two complex reimbursement landscapes simultaneously, whereas separate brands could be optimized for each. The practical consequence was that a patient whose insurance covered smoking cessation pharmacotherapy but imposed restrictions on psychiatric medication could access Bupropion as Zyban in a way they might not have been able to access it as Wellbutrin. This is either a story about insurance company classifications creating unnecessary complexity, or a story about pharmaceutical companies successfully navigating that complexity to get medication to patients who needed it. Probably both. The patient-facing confusion this arrangement produced was real and recurring. People noticed. A patient whose physician had switched them from Wellbutrin to Zyban, while their dose stayed the same, and their tablet looked identical, would understandably ask questions. Patients whose partners were taking Zyban for smoking, and who were themselves taking Wellbutrin for depression, sometimes had conversations that required physicians to explain, with appropriate nuance, that yes, the medications were the same, and no, that did not mean what they might initially think it meant. Those conversations were awkward in ways that the pharmaceutical industry had presumably anticipated and decided to manage rather than prevent. The molecule did not care what it was called. The humans involved cared quite a lot. The mechanism by which Bupropion exerts its therapeutic effects is real, reasonably well characterized, and simultaneously more complicated than the summary version. It inhibits the reuptake of dopamine and norepinephrine, fully captures. This is true of most drugs to some degree. The summary version is always a simplification of a biological process that does not simplify easily. But with Bupropion, the gap between the summary and the full picture is wider than average, in ways that matter for understanding why the drug works the way it does, why it works differently for different people, and why its clinical profile has the specific character that distinguishes it from other antidepressants. The reuptake inhibition story is accurate as far as it goes. Bupropion blocks the dopamine transporter and the norepinephrine transporter, the proteins embedded in neuronal membranes that, after a neurotransmitter has been released into the synapse and done its signaling work, pull it back into the neuron for recycling. By blocking these transporters, Bupropion extends the time that dopamine and norepinephrine remain active in the synapse, increasing the amplitude and duration of the signal they produce at postsynaptic receptors. This is the same broad mechanism as the stimulants. Cocaine blocks the dopamine transporter, amphetamine both blocks it and causes reverse transport. But Bupropion executes it at substantially lower potency and through pharmacokinetics that produce a gradual sustained effect rather than an acute one. Cocaine reaches peak plasma concentrations within minutes of administration and produces a dopamine surge that is rapid, intense, and brief. Bupropion, in its extended-release formulation, reaches peak plasma concentrations over several hours and produces a modest, sustained increase in dopamine and norepinephrine availability that develops gradually over weeks of treatment. These are not simply quantitative differences, they are qualitative differences in the pattern of dopaminergic signaling, and those differences are pharmacologically significant in terms of both the therapeutic effect and the addiction liability. Bupropion's dopamine transporter affinity is relatively modest, substantially lower than cocaine's and lower than most synthetic cathinones in the bath salts class. Its norepinephrine transporter affinity is higher, and some researchers argue that the norepinephrine component contributes more to the antidepressant effect than the dopamine component, with the dopamine component contributing more to the motivational and processation effects. This differential, the norepinephrine-weighted mechanism, helps explain why Bupropion's clinical profile overlaps partially with other norepinephrine-active antidepressants while remaining distinct from them. There is a third mechanism that became clearer in later research. Bupropion is also a nicotinic acetylcholine receptor antagonist. It blocks specific subtypes of nicotinic receptors, the receptors that nicotine acts on, with a potency that is clinically significant at therapeutic plasma concentrations. This nicotinic antagonism appears to contribute to both the smoking cessation effect by reducing the rewarding properties of nicotine and possibly to some aspects of the antidepressant mechanism through pathways that involve the interaction between cholinergic and dopaminergic systems. The nicotinic component was not part of the original mechanistic description of bupropion and was characterized substantially after the drug was already in clinical use, a reminder that the understanding of a drug's mechanism can continue to deepen long after the drug is approved and in wide use. The metabolites of bupropion add another layer of complexity. Hydroxybupropion, the primary metabolite, reaches higher steady-state plasma concentrations than bupropion itself, and has its own pharmacological activity at monoamine transporters and nicotinic receptors. The experience of taking bupropion is, pharmacologically, the combined experience of bupropion and its active metabolites in proportions that vary by individual, by dose, and by the genetic variants that determine metabolic rate. Understanding this helps explain why the drug's effects vary more between individuals than its mechanism might suggest. The variation in what people experience is not random noise, but reflects genuine differences in the pharmacokinetic picture each person's body produces from the same starting compound. There is a quality of clinical observation that shows up consistently enough in patient reports and practitioner experience to deserve serious attention even before it is fully explained at the level of mechanism, and with bupropion, that observation is this: it feels different. Not just chemically different from the SSRIs, but experientially different, in ways that patients reliably describe with a specific vocabulary that recurs across contexts, cultures, and clinical settings, often enough to constitute meaningful signal. The SSRI experience, in patient descriptions that appear repeatedly across clinical literature, memoirs, online communities, and the accumulated informal record of a drug class that has been widely prescribed for over 3 decades, tends to be described in terms of regulation. The sharp edges are softer, the lowest points are higher, the anxiety is quieter, the rumination loses some of its grip. The emotional landscape becomes more navigable, not flat, not blunted, but less violent in its oscillations. For many patients, this is exactly the therapeutic effect that was needed, and the SSRIs genuinely deliver it. The emotional regulation they provide is a real and valuable clinical outcome, but some patients describe a different experience. The negative frequencies are quieter, they say, but so are some of the positive ones. The dark thoughts come less often, but the light ones seem further away too. The floor has been raised, but the ceiling has also come down. Getting through the day is easier, but wanting to do anything in particular with the day, feeling the forward pull of goals and activities that had once seemed worth pursuing, is not fully restored. This is not a universal experience with SSRIs, and for many patients it does not apply at all. But it is a consistent enough report to have a name in the clinical literature: emotional blunting, or SSRI-associated apathy, and it appears to be a real phenomenon with a biological substrate related to the serotonin system's complex role in both emotional regulation and motivational engagement. Bupropion, in contrast, is more often described in terms of activation, more energy, more forward momentum, the return of something that had been absent, not just the reduction of something bad, but the restoration of something that should have been there. Patients describe being able to start things again, to want things again, to feel that getting up and doing something has a point, that effort connects to outcome, that the future is worth turning toward. The clinical term for what is being restored is motivation, the capacity to initiate and sustain goal-directed behavior, and its restoration feels qualitatively different from simple mood improvement. The dopamine mechanism is the most persuasive explanation for this difference. Dopamine in the prefrontal cortex and the nucleus accumbens is centrally involved in incentive salience, the property of making goals feel worth pursuing, of generating the pull toward anticipated rewards, of creating the subjective experience that something is worth doing. This is distinct from the hedonic impact of reward, the pleasure of the actual experience, and it is this anticipatory motivational dimension that appears to be selectively reduced in the energic, anhedonic presentations of depression that respond particularly well to bupropion. The practical clinical implication is that bupropion tends to work best, or works in a way that is specifically different from the SSRIs, for patients whose depression is dominated by motivational shutdown, energy deficit, and the inability to experience anticipatory pleasure. For patients whose depression looks more like anxiety, rumination, and emotional volatility, the activation profile of bupropion can be counterproductive, adding energy to a system that is already running too fast, exacerbating the restlessness and irritability that are already present. This differential has direct implications for clinical matching, for the practical question of which patients are most likely to benefit from bupropion specifically, as opposed to benefiting from any antidepressant treatment. The dopamine difference is also the structural difference. Bupropion engages the dopamine system because its cathinone scaffold is well suited to interacting with the dopamine transporter in a way that the SSRIs, which sit on a different molecular scaffold with different transporter selectivity, do not. The clinical difference and the chemical difference are the same difference, viewed from different scales. The story of synthetic cathinones as recreational drugs is, at its core, a story about a specific legal vulnerability that existed in drug regulatory frameworks for several decades, a gap between the rate at which novel compounds could be synthesized and characterized, and the rate at which regulatory systems could evaluate, schedule, and prohibit them. It is a story about what happens when chemistry is accessible. The internet removes geographic constraints on commerce, and the commercial incentive to find legal alternatives to controlled substances is strong enough to sustain an industry built specifically on staying one structural modification ahead of the law. The legal gap was structural and, in retrospect, almost inevitable. Drug laws in most jurisdictions regulate specific named compounds. The Controlled Substances Act in the United States schedules specific molecules: amphetamine, methamphetamine, cocaine, cathinone, by name and by specific structural definition. A law that schedules cathinone does not automatically schedule every possible structural variant of cathinone, because the law cannot preemptively name compounds that have not yet been synthesized. The law was designed for a world in which new psychoactive compounds arrived rarely and through identifiable pharmaceutical channels. It was not designed for a world in which chemists could synthesize, characterize, and bring to market dozens of new structural variants within months. The Federal Analog Act of 1986 in the United States attempted to address this gap by creating a mechanism for treating unscheduled compounds as Schedule I substances if they were substantially similar to a scheduled substance and intended for human consumption. The Act was conceptually sound. It recognized the gap and tried to close it, but its application in practice was complicated by two significant limitations. The first was the substantially similar standard, which required the government to establish structural and pharmacological similarity in each case, effectively requiring a case-by-case scientific argument rather than a categorical prohibition. The second was the intended for human consumption requirement, which the synthetic cathinone industry addressed with the straightforward tactic of labeling their products as not for human consumption, a legal fiction so transparent that it became its own commentary on the gap between regulatory intent and regulatory implementation. but that nevertheless created sufficient legal uncertainty to complicate enforcement. In Europe, where regulatory frameworks varied significantly across different national jurisdictions, the gap was navigable in different ways in different countries. The European Monitoring Centre for Drugs and Drug Addiction tracked the emergence of new synthetic cathinones through its early warning system. But tracking is not the same as scheduling, and scheduling in individual member states followed different timelines and different legal processes. A compound banned in the United Kingdom might remain legal in another European jurisdiction, and internet commerce meant that legal status in one country was not the practical barrier it would have been in a pre-internet market. The synthetic cathinone industry that developed within this gap was not primarily composed of sophisticated criminal organisations. It was, at least in its early years, primarily composed of chemists and entrepreneurs operating in legal grey areas. People who were aware of the regulatory landscape were deliberately designing products to navigate it, and were selling to an adult market that was seeking legal alternatives to controlled stimulants. The moral evaluation of this enterprise is complicated by its eventual consequences, which included significant harm from compounds that were brought to market without adequate toxicological characterization. But understanding what it was, a legal industry exploiting a structural gap, not primarily a criminal one evading clear prohibition, matters for understanding why the regulatory response took as long as it did and why it took the forms it did. Bupropion, sitting across the pharmaceutical divide from all of this, continued to be prescribed and dispensed through regulated channels throughout this period. The chemistry connecting it to the synthetic cathinone world was real and visible to anyone who looked at the structures. The social worlds were entirely separate. The products that became known colloquially as bath salts were primarily synthetic cathinones, compounds built on the cathinone scaffold with specific substitutions designed to produce stimulant effects while remaining, at least temporarily, outside the scope of existing drug schedules. The name itself is a retail artifact. These substances were sold in head shops, gas stations, and online retailers in small foil packets labelled as bath salts, plant food, or research chemicals, with not for human consumption printed on the packaging in the same spirit that a note on a cake reading not for eating inspires confidence. The labelling was a legal shield, not a description. Everyone involved understood this, including the customers, the retailers, and eventually the regulators. The three compounds most closely associated with the bath salts phenomenon in the United States and Europe were mephedrone, methylone, and methylene dioxypyrovalerone, MDPV. Each had a distinct pharmacological profile, and understanding the differences between them matters for understanding both the range of experiences associated with bath salts use and the range of risk that the category contained. Mephedrone, street names including Meow Meow and MC, chemical name 4-methylmethcathinone, is a substituted cathinone that affects the serotonin, dopamine, and norepinephrine systems in a pattern that produces effects broadly comparable to a combination of amphetamine and MDMA. Euphoria, increased sociability, elevated energy, heightened sensory experience, and a duration of effect of approximately 3 to 5 hours. It emerged as a significant recreational drug in the United Kingdom around 2007, spread rapidly across Europe through a combination of online sales and word of mouth, and was used extensively at music events and in social settings where MDMA and amphetamine had previously been the primary choices. The United Kingdom classified it as a Class B controlled substance in 2010, the same classification as amphetamine, following several high-profile deaths in which mephedrone was involved, though not always causally implicated. Methylone is structurally similar to MDMA. It shares the methylene deoxy ring modification that characterizes the ecstasy family, with the addition of the beta-keto group that makes it a cathinone. Its effects are described by users as broadly similar to MDMA, but with a shorter duration and a somewhat different quality of empathogenic effect. It was one of the compounds commonly found in products sold as bath salts and Molly in the United States, and its presence in products that users believed contained MDMA created a layer of additional risk through undisclosed substitution. MDPV, methylene dioxypyrovalerone, is the compound most closely associated with the severe toxicity reports that drove the bath salts panic. It is a potent dopamine transporter blocker, substantially more potent at the DAT than cocaine, with relatively weaker effects on the serotonin and norepinephrine systems. Its pharmacological profile produces intense stimulation, a very short subjective duration of effect combined with intense craving for redosing, and at higher doses paranoid psychosis characterized by the kind of agitated, hypervigilant, reality-detached state that produced the behavioral descriptions in media coverage. The compulsive redosing pattern, driven by the short perceived duration and intense craving, is pharmacologically consistent with high-potency dopamine transporter blockade and represents a significantly higher addiction liability than either mephedrone or methylone. All three of these compounds are structural relatives of bupropion. All three share the cathinone scaffold. The differences between bupropion and the most dangerous of them, MDPV, represent the full range of what substitution chemistry can do to a shared molecular core, from a modest, sustained, therapeutically useful dopamine modulation to an intense, acute, highly reinforcing dopamine flooding that drove some of the most alarming clinical presentations in recent drug toxicology. Same family, profoundly different members. The scaffold is the beginning of the story, not the end of it. In the early months of 2012, synthetic cathinones achieved a level of mainstream media attention that was, measured against the actual epidemiology of their use, dramatically disproportionate to the pharmacological reality. The coverage was not fabricated, the harms were real, the incidents were real, and the compounds involved were genuinely dangerous in ways that warranted public health concern. But the story that media coverage constructed around those real events was inflated, distorted, and filtered through a template of drug panic that American journalism had been applying with remarkable consistency to new psychoactive substances since at least the 1930s. If you have seen this movie before, and if you're old enough to remember the crack cocaine coverage of the 1980s, or the methamphetamine coverage of the early 2000s, you will recognize the structure immediately. The triggering event for the most intense period of coverage was a series of incidents in which individuals behaved violently or self-destructively while under the influence of substances reported to contain synthetic cathinones. The incidents were reported with a level of graphic specificity and dramatic emphasis that transformed them from toxicological events into cultural symbols. The phrase zombie drug appeared in headlines across the country. Reports described users as having extraordinary physical strength, being impervious to pain, and engaging in behaviours so extreme that the implication was clear. This substance was doing something unprecedented to the human brain, something outside the normal pharmacological range, something that required new vocabulary to describe. Toxicologists and emergency medicine physicians found themselves in the uncomfortable position of trying to correct a narrative that had already achieved cultural velocity. The pharmacological reality they were attempting to communicate was both accurate and, in the context of a news cycle that had already committed to the zombie framing, essentially inaudible. What they were saying was this: synthetic cathinones produce stimulant toxicity, a well-characterized clinical syndrome involving elevated heart rate, elevated blood pressure, hypothermia, agitation, and at high doses paranoid psychosis. The specific behavioural presentations being described in media coverage were consistent with severe stimulant toxicity and with the specific pharmacological profile of high-potency dopamine transporter blockers like MDPV. They were not consistent with the introduction of a new pharmacological mechanism that had never been seen before. They were an extreme version of something that toxicologists and emergency physicians had been treating for decades. The compounds were new. The syndrome was not. What distinguished the synthetic cathinone toxicity presentations from typical amphetamine or cocaine toxicity was primarily the potency of the dopamine transporter blockade in the most dangerous compounds, particularly MDPV, and the specific pattern of compulsive redosing that this potency encouraged. A person who'd been awake for 3 days, repeatedly redosing a high-potency dopamine transporter blocker in response to intense craving, with escalating plasma concentrations and a brain that had been in a state of dopaminergic overstimulation for an extended period, would present to an emergency department in a state of acute psychosis that was genuinely alarming to treat and that required specific management protocols. This was a real clinical problem. It was not a supernatural one. The most famous incident driving the media coverage, a violent attack in Miami in May of 2012, was widely attributed to bath salts in initial reporting. Subsequent toxicological analysis found only marijuana in the victim's system. This correction received a fraction of the coverage that the original attribution had generated, which is precisely the pattern that characterizes drug panics. The alarming claim spreads rapidly, the correction spreads slowly, and the cultural impression formed by the alarm persists long after the specific factual basis for it has been revised. The regulatory response was swift, partly because the media attention created political pressure that demanded legislative action, and partly because there were genuine public health grounds for scheduling the specific compounds involved. The Synthetic Drug Abuse Prevention Act of 2012 placed numerous synthetic cathinones on Schedule 1. Emergency scheduling had already been used against specific compounds in the preceding months. The combination of media attention, political pressure, and genuine public health concern produced legislative action on a timeline significantly faster than the normal scheduling process would have allowed. The underlying structural challenge, that novel compounds could be synthesized faster than they could be scheduled, was not resolved by the 2012 legislation. It was managed temporarily by the specific compounds covered. Manufacturers responded, as they had throughout the legal high era, by modifying their products to incorporate compounds not covered by the new schedules. The cat and mouse dynamic continued. The specific products changed. The structural gap that had allowed the industry to exist in the first place remained. Bupropion, throughout all of this, continued to be dispensed by pharmacists in amber pill bottles to people managing depression and nicotine dependence. The pharmacists did not modify their dispensing practices in response to the bath salts panic. The physicians did not alter their prescribing of Wellbutrin. The regulatory agencies did not revisit the approval of Bupropion in light of its structural relationship to the scheduled compounds, because the structural relationship, while real, was not evidence of equivalent risk, and the distinction between a compound's molecular ancestry and its clinical profile is precisely the kind of distinction that a functioning regulatory system exists to make and maintain. From a purely structural standpoint, which is, let's be clear, not the only standpoint that matters, but is the one that makes the Wellbutrin connection legible, the relationship between Bupropion and the synthetic cathinones that made headlines in 2012 is not subtle. Place the molecular structures side by side and the shared scaffold is immediately apparent. The phenethylamine backbone, the beta-keto group at the beta-carbon, the amino group at the terminus, the cathinone skeleton is present in all of them, in the same position, doing the same structural work. They are members of the same molecular family in the most literal chemical sense. Their family resemblance is as real and as visible as any family resemblance gets in organic chemistry. What differs is everything attached to that core, and in medicinal chemistry, everything attached to the core is where the pharmacology lives. Bupropion's specific substitutions, the tertiary butyl group on the nitrogen and the chlorine at the para position of the aromatic ring, were not chosen arbitrarily. The tertiary butyl group is large and sterically bulky, and its presence on the amino nitrogen substantially reduces the compound's affinity for the dopamine transporter. Affinity is the key variable. It determines how tightly and how specifically a compound binds to its target protein, and in the context of dopamine transporter pharmacology, lower affinity means weaker inhibition, which means a more modest and more gradual effect on dopamine availability in the synapse. This is precisely what distinguishes Bupropion's therapeutic profile from the more potent cathinones. Not a different mechanism, but the same mechanism operating at a substantially lower intensity. Compare Bupropion's dopamine transporter affinity to that of MDPV, methylenedioxypyrovalerone, the compound most associated with severe synthetic cathinone toxicity. MDPV has a pyrrolidine ring on its nitrogen, a 5-membered cyclic amino group that is geometrically very different from Bupropion's tertiary butyl group, and a methylenedioxy bridge on the aromatic ring. These modifications produce a compound with dramatically higher dopamine transporter affinity, estimated in some studies at several times the potency of cocaine. The same transporter, the same basic mechanism, an intensity difference large enough to produce entirely different clinical and toxicological outcomes. The structure-activity relationship within the cathinone family has been characterized in remarkable detail, partly through academic pharmacology research, partly through the pharmaceutical research conducted around bupropion and related compounds, and partly in a contribution that is scientifically valuable, even if it arrived through channels that regulators found problematic, through the systematic documentation of the broader phenethylamine family in Alexander Shulgin's PCAL. The accumulated structure-activity data allows medicinal chemists to predict, with reasonable accuracy, how modifications to the cathinone scaffold will affect dopamine transporter affinity, norepinephrine transporter selectivity, serotonergic activity, and pharmacokinetic properties. This predictive capacity is one of the reasons the cathinone scaffold remains an active area of pharmaceutical research, despite its complicated cultural associations. The family tree metaphor is useful up to a point, and it is worth being specific about where it becomes misleading. Family trees imply shared characteristics across generations, the idea that if you know one member of a family, you know something meaningful about the others. In pharmacology, the cathinone family does share a fundamental characteristic: the ability to engage monoamine transporter systems in ways that affect dopamine, norepinephrine, and serotonin availability. That is the family characteristic. But the range of expression of that characteristic, from Bupropion's modest, therapeutic, well-tolerated modulation to MDPV's intense, rapidly reinforcing, acutely toxic profile, is wide enough that knowing one member tells you relatively little about any specific other member without knowing the specific substitutions. The comparison that gets made, sometimes in good faith, sometimes to generate alarm, between Bupropion and bath salts is accurate at the level of scaffold and inaccurate at the level of pharmacological profile. They share an ancestor. They are not equivalent. A chemist who understood the structure-activity relationships would not be surprised by the connection and would not be alarmed by it. A journalist who noticed it without the pharmacological context might reasonably find it striking. The difference in reaction is a difference in the depth of understanding available, not a difference in what the molecules actually are. No honest account of how we came to understand substituted cathinones and substituted phenethylamines generally can avoid mentioning Alexander Shulgin. He was a chemist whose career defied easy categorization and whose contributions to the field were as significant as they were controversial. He was not a fringe figure operating in the shadows of legitimate science. He was a credentialed, licensed, peer-reviewed scientist who happened to be interested in a category of compounds that the establishment found deeply uncomfortable, and who pursued that interest with a rigour and transparency that was either admirable or dangerous depending on who you asked, and which specific aspect of the work you were evaluating. Shulgin was born in 1925 in Berkeley, California. He studied at Harvard, left to serve in the Navy, returned to complete his education, and received his doctorate in biochemistry from UC Berkeley in 1955. His early career took him to Dow Chemical, where he developed a commercially successful pesticide, a contribution that earned him a degree of scientific autonomy that he used to pursue interests the company might otherwise have discouraged. He synthesized compounds in his personal laboratory, reported their effects through channels that were not standard pharmaceutical protocols, and eventually left Dow to pursue his research independently. His formal relationship with the DEA was, for several decades, surprisingly collaborative. He held a Schedule I research license, a license permitting work with controlled substances for legitimate research purposes, and he provided expert consultation to the DEA on chemical analysis and forensic identification of controlled substances. He was, by the DEA's own operational account, a useful scientific resource. He understood the chemistry of psychoactive compounds more thoroughly than almost anyone alive, and that understanding was professionally valuable to law enforcement agencies trying to identify novel substances appearing in drug markets. The tension in this relationship was always present and eventually became irreconcilable. Shulgin's research program was not oriented toward law enforcement applications. It was oriented toward understanding the relationship between molecular structure and psychoactive effect, toward mapping the pharmacological landscape of the phenethylamine family with the same systematic rigour that a naturalist might apply to mapping a previously undescribed ecosystem. He synthesized compounds, characterized their chemistry, and documented their subjective effects through a process of direct self-experimentation conducted with a small group of research associates who participated voluntarily and with full awareness of what they were doing. The results of this research were published in 2 books, Pichkal, Phenethylamines I Have Known and Loved, in 1991, co-written with his wife Anne Shulgin, and Tichkal, Tryptamines I Have Known and Loved, in 1997. These books are unlike any other scientific publications. They combine autobiography, pharmacology, synthetic chemistry, philosophy, and love story in a form that has no precedent in the scientific literature. They document the synthesis and effects of hundreds of compounds in unprecedented detail. They are simultaneously the most comprehensive reference works available on phenethylamine and tryptamine pharmacology, and the publications that most clearly illustrate the tension between scientific knowledge and regulatory control. Pichkal specifically covers the substituted phenethylamines, including cathinone-related compounds, and its documentation of the structure-activity relationships within this family is directly relevant to understanding the pharmacological position of Bupropion within its molecular family. Shulgin's work characterized the effects of cathinone itself and of various structural modifications, providing the foundation of understanding that subsequent academic research has built on. His contribution to the pharmacological knowledge base that allows us to understand why bupropion is different from MDPV, and why both are members of the same structural family, is substantial. The DEA eventually revoked his Schedule I research license following an inspection of his laboratory in 1994. an action widely understood as a response to the publication of Percal and the potential for its detailed synthesis information to function as a practical guide. Shulgin's position that chemistry is not inherently dangerous, that knowledge is better than ignorance, and that the direct study of psychoactive effects on human consciousness is legitimate science, was a principled stance that the regulatory framework could not accommodate. He died in 2014 at 88, having done more to characterize the pharmacological landscape of the phenethylamine family than any other single scientist. His work is cited in academic literature, consulted by forensic chemists, and sits in an uncomfortable but undeniable relationship with both the pharmaceutical history of Bupropion and the regulatory history of synthetic cathinones. The arc of Bupropion's history is, in miniature, the story of how pharmaceutical development actually works, not the polished version presented in regulatory submissions and pharmaceutical advertising, but the actual version with all its accidents, setbacks, unexpected discoveries, near endings, and structural dependence on observations that nobody planned to make and outcomes that nobody fully anticipated. The compound was synthesized in 1969 by a medicinal chemist working in a systematic exploration of a chemical family. Its mechanism, dopamine and norepinephrine reuptake inhibition, was a consequence of which scaffold the researchers were working on, not the result of a targeted design process aimed specifically at those transporters. Its initial approval in 1985 was followed by a safety signal that the clinical trials had not predicted, because the vulnerable population had not been adequately represented in the pre-approval data. Its voluntary withdrawal addressed the safety problem by demonstrating institutional responsibility in a way that the regulatory alternative, compelled withdrawal, would not have permitted. Its reformulation solved the technical problem that had generated the safety signal. Its reapproval positioned it to take advantage of an application nobody had planned for. And its most commercially significant indication, smoking cessation, was discovered by physicians noticing something unexpected in patients they were treating for something else entirely. Every step in this chain is contingent. Remove any one of them, and the drug's history changes substantially. If the clinical trial population had included more patients with eating disorders, the seizure risk might have been characterized pre-approval rather than post-marketing, which could have led to earlier reformulation, earlier reapproval, and a different commercial trajectory in the critical window when the SSRIs were establishing dominance. If Burroughs' Wellcome had responded to the post-marketing seizure reports with denial rather than voluntary withdrawal, the regulatory response would likely have been more aggressive and the subsequent development pathway more difficult. If the physicians noticing the smoking effects had not recorded them, or had not reported them to the company, the smoking cessation indication might have been discovered years later or not at all. The history we have is one path through a landscape that contained many other possible paths. This contingency is not specific to Bupropion. It characterizes drug development generally, and recognizing it is important for understanding what drug development can and cannot reliably produce. The process is not a machine that takes clinical needs as inputs and reliably produces medications as outputs. It is a human process that is responsive to clinical needs, constrained by scientific understanding and technical capability, shaped by commercial incentives and regulatory requirements, and dependent at critical moments on the quality of observation, the depth of institutional patience, and the willingness to follow unexpected results rather than discarding them because they do not fit the existing plan. The Bupropion story also illustrates the specific pharmacological principle that structural families in chemistry carry pharmacological implications across their members. The cathinone scaffold that Bupropion shares with both the khat plant's active compound and the synthetic cathinones of the bath salts era is not a coincidental resemblance. It is the chemical reason that all of these compounds interact with monoamine transporter systems. The scaffold determines the family of biological activity. The specific substitutions determine the intensity, selectivity, and clinical character of that activity within any individual compound. Understanding this principle is not merely academically interesting. It has practical implications for how pharmaceutical research approaches novel compound development, how regulatory agencies evaluate new substances appearing in drug markets, and how clinicians understand the mechanistic basis of the drugs they prescribe. A physician who understands that bupropion is a substituted cathinone is not necessarily more alarmed by prescribing it. The clinical evidence for its safety and efficacy is not changed by knowing its structural family, but they are better equipped to understand its pharmacological profile, to anticipate the kinds of drug interactions that structural relationships can predict, and to contextualize both the drug's effects and its limitations within a framework that is more complete than the summary mechanism alone provides. Here, at the end of the structural and historical story, is the thing that tends to produce a specific pause when people first encounter it. The Wellbutrin and bath salts connection does not change anything practical. It does not alter how bupropion works as a medication. It does not change its safety profile, its efficacy for depression or smoking cessation, or the risk-benefit assessment that supports its continued clinical use. It does not mean that taking a prescribed antidepressant is pharmacologically equivalent to taking a recreational stimulant. The distances within the cathinone family are real, and they are large. What it does is illuminate something important about chemistry, about pharmacology, and about the human tendency to sort molecules into moral categories that the molecules themselves do not observe. And that illumination is worth sitting with, because it reveals something about how we understand drugs, all drugs, not just this one, that tends to be obscured by the categorical thinking that drug policy and pharmaceutical marketing both encourage for their own reasons. Chemistry does not respect the categories we impose on it. The cathinone scaffold exists simultaneously in an FDA-approved antidepressant taken by tens of millions of people, in a plant used as a traditional social substance by tens of millions of other people over centuries, in compounds scheduled as Schedule I controlled substances with no accepted medical use, and in synthetic recreational drugs that produced legislative emergencies in multiple countries. The scaffold does not know what category it is supposed to be in. It interacts with monoamine transporter proteins because that is what the structure is geometrically suited to do, and the legal status, the brand name, the clinical indication, and the cultural context are human constructions layered around the chemistry, not properties of the chemistry itself. This is not an argument for regulatory nihilism, or for treating all cathinone-derived compounds as equivalent. The differences between bupropion and MDPV are pharmacologically real, clinically significant, and fully justify the different regulatory treatments they receive. A compound with bupropion's potency, selectivity, and pharmacokinetic profile belongs in a different regulatory category from a compound with MDPV's profile, and placing them in different categories is not arbitrary. It reflects a genuine difference in the risk-benefit balance that each compound presents. The regulatory system, in this instance, is doing approximately what it should be doing. The deeper point is about the nature of pharmacological knowledge and its relationship to the cultural stories we build around drugs. Bupropion is understood as a safe, effective, unremarkable antidepressant, a standard of care medication that physicians prescribe routinely and patients take daily without anxiety. Bath salts are understood as a dangerous, alarming category of substances that generated a national panic and required emergency legislation. These understandings are not wrong. They are accurate representations of the clinical and public health realities of the two categories, but they are built on a shared chemical foundation that the stories do not acknowledge, and acknowledging it produces a more complete and more honest picture of what these compounds actually are. The picture is this: the same molecular scaffold in the hands of a pharmaceutical company with clinical trial infrastructure and regulatory oversight produces a medication. In the chemistry of a plant that has been part of human culture for centuries, it produces a traditional social substance. In the hands of chemists operating in legal grey zones without toxicological oversight or controlled dosing, it produces compounds that harm people. The scaffold is consistent throughout. The human context is what varies, and the human context is what determines whether the outcome is a prescription, a cultural practice, or a public health emergency. This is the story of the pill, the plant and the panic. It is chemical and historical and cultural all at once, and it ends where most good pharmacological stories end, not with a clean conclusion, but with a more precise understanding of a complicated thing, which is the closest that chemistry and human beings together tend to get to resolution. Most people, when they take a medication, think of it as a single thing. You swallow the pill. The pill does its job. You feel better, or you don't. The chemistry is somewhere in the middle, doing whatever chemistry does, invisible and unnarrated, like the backstage crew at a theatre production that everyone pretends doesn't exist because acknowledging it would complicate the experience of watching the show. But what happens to bupropion after it enters the body is considerably more interesting than the summary version suggests, and it matters for understanding why different people have such different experiences with the same drug at the same dose, why the relationship between dose and effect is non-linear in ways that required reformulation to manage, and why the clinical profile of bupropion has the specific character that distinguishes it from other antidepressants. The journey begins in the gastrointestinal tract. Bupropion is absorbed through the gut wall and enters the bloodstream, from which it crosses the blood-brain barrier with relative ease because it is lipophilic enough to partition into fatty tissue, a structural property that is, not coincidentally, a consequence of the cathinone scaffold and the specific substitutions that make it bupropion rather than something more water-soluble. The extended-release formulations, the ones currently in standard clinical use, slow this absorption by embedding the compound in a matrix that releases it gradually as the tablet moves through the intestinal tract, flattening the peak plasma concentration curve that the immediate release formulation produced so sharply and so dangerously. Once in the bloodstream, bupropion is distributed throughout the body and brain, and it begins to be metabolized. The primary metabolic pathway involves an enzyme called CYP2B6, one of the cytochrome P450 enzymes that the liver uses to process foreign compounds and prepare them for excretion. The CYP450 system is the liver's primary chemical processing facility, and CYP2B6 is one of its more variable components, meaning that the gene encoding it differs significantly between individuals in ways that affect metabolic rate. Some people have 2 functional copies of the gene and metabolize bupropion at a standard rate. Some have 2 loss-of-function copies, poor metabolizers, and metabolize it much more slowly, leading to accumulation to higher plasma concentrations at any given dose. Some are ultra-rapid metabolizers who clear it so quickly that standard doses may not maintain therapeutic plasma concentrations. This genetic variability in metabolism is clinically significant in several ways that are not always appreciated in prescribing practice. A poor metabolizer taking the standard dose of 150 mg of Wellbutrin XL is, in pharmacokinetic terms, receiving a higher effective dose than a standard metabolizer taking the same tablet. The nominal dose is the same. The exposure, the area under the plasma concentration time curve, is substantially higher. and since the seizure risk, the cardiovascular effects, and the potential for activating side effects like insomnia and anxiety are all dose-dependent, poor metabolizers are at higher risk for these effects at standard doses than their prescribing information alone would suggest. The metabolites of bupropion are also pharmacologically active, which adds a layer of complexity that is not always acknowledged in clinical discussions. The primary metabolite, hydroxybupropion, in its threo- and erythrodiesteromeric forms, reaches steady-state plasma concentrations that are substantially higher than the parent compound itself, and it has its own pharmacological activity at norepinephrine and dopamine transporters and at nicotinic acetylcholine receptors. What a patient experiences when they take bupropion is not purely the effect of bupropion, it is the combined effect of bupropion and its metabolites, in proportions that vary based on individual metabolic phenotype. This metabolic complexity helps explain one of the most clinically notable features of bupropion, the significant individual variation in response. Patients taking the same dose of the same formulation report experiences that differ substantially in the degree of activation, the quality of mood effect, the presence and severity of side effects, the time to onset of benefit. This variation is not purely placebo effect or subjective reporting noise. Much of it reflects real differences in the pharmacokinetic picture that different individuals' bodies produce from the same starting compound. Understanding this is useful for both prescribers and patients. It provides a mechanistic framework within which this drug works differently for different people, is not a clinical mystery, but a predictable consequence of metabolic genetics. Given that bupropion is a dopamine reuptake inhibitor, given that it engages the same transporter that cocaine and amphetamine engage, in a mechanistically similar way, the question of whether it has addiction liability is entirely reasonable to ask, and it deserves a careful, accurate answer rather than a dismissive wave toward the pharmaceutical company's assurances. Because the question is not silly. The mechanism is real. The concern is logical. The answer is just more nuanced than a yes or a no. The answer, carefully stated, is this. Bupropion has very low addiction liability in the context of therapeutic use, for reasons that are mechanistically explicable and empirically supported by decades of post-marketing surveillance data. But very low is not zero, and the reasons matter for understanding both the drug's actual safety profile and the pharmacological principles that govern addiction liability more broadly. The addiction liability of a dopaminergic compound is determined primarily by the speed and magnitude of the dopamine signal it produces in the brain's reward circuitry. This is not a hypothesis. It is one of the most robustly supported findings in addiction neuroscience, replicated across compounds, animal models, and human studies for several decades. Cocaine produces a fast, intense surge of dopamine in the nucleus accumbens by blocking the dopamine transporter with high affinity and rapid kinetics. The brain's reward learning machinery, which is exquisitely sensitive to the timing and magnitude of dopamine signals, encodes this surge as highly salient, worth repeating, worth seeking out. The addiction is the learned behavior that results from that encoding process, reinforced by every subsequent dose. Bupropion inhibits the dopamine transporter substantially more weakly than cocaine. Its DAT affinity is roughly 10 times lower. More importantly, its pharmacokinetics, particularly in the extended-release formulation, produce a slow, gradual increase in dopamine availability that develops over hours rather than minutes, and that reaches a modest plateau rather than a sharp peak. The brain's reinforcement learning system responds to peaks, to rapid, sharp increases in dopamine, not to slow, gradual elevations. Bupropion, by producing the latter, does not engage the reinforcement machinery in the way that fast, potent dopaminergic compounds do. The signal is there. It is just speaking at a volume and a speed that the reinforcement system does not find particularly compelling. This explanation is supported by clinical data. The abuse potential of bupropion has been evaluated in formal human laboratory studies using measures validated against known drugs of abuse. In subjects with histories of recreational stimulant use, a population specifically selected because they would be more likely than the general population to find dopaminergic compounds rewarding, bupropion at therapeutic doses produces weak or absent signals on measures of drug liking, desire for more, and euphoria. The subjective stimulant effects are present but modest. The reinforcing value as measured by standard behavioral pharmacology methods is low. Reports of bupropion misuse exist in the clinical literature. Cases of people crushing extended-release tablets and insufflating the powder to defeat the slow-release mechanism and achieve a faster, more intense effect. These reports are important because they illustrate that the addiction liability of bupropion in standard use is substantially a function of its slow oral pharmacokinetics, rather than purely a function of its intrinsic DAT affinity. Administer the same total dose by a route that produces rapid brain exposure, and the addiction liability increases correspondingly. This is consistent with the pharmacological principles governing abuse potential across all compounds. It is not a special property of bupropion but a demonstration of the general principle that route and speed of administration modulate reinforcing value independent of intrinsic potency. The clinical conclusion is that bupropion, prescribed and used as directed, has an addiction liability that is clinically negligible for the vast majority of patients. The structural relationship to cocaine and the synthetic cathinones is real. The practical implication for prescribed therapeutic use is minimal. Both of those statements can be true simultaneously, because they are describing different aspects of the same pharmacological story. And so here we are. You have travelled tonight from a laboratory in 1969 to a tuberculosis ward where patients started dancing. From a plant growing quietly in the highlands of East Africa to a pharmaceutical company in London with patience enough to follow a molecule for 2 decades without knowing where it would lead. From a small white pill in an amber bottle to the strangest structural family reunion in the history of pharmacology. You've learned that the brain is not a simple machine with simple levers, that the same scaffold can be a medication and a menace depending on who is holding it and why. That accidents produce discoveries, that discoveries produce more questions, and that the distance between a prescription and a panic is, more often than not, a matter of context rather than chemistry. The people who synthesized bupropion were not trying to change the world. They were trying to do the next careful thing. The physicians who noticed their patients were quietly putting down their cigarettes were not running experiments. They were paying attention. The patients who described the return of motivation as feeling like themselves again were not reporting pharmacokinetics. They were reporting the experience of being human, restored. That is what medicine is at its best. Not a system of molecular interventions administered to biological machines, but a conversation between chemistry and human experience conducted imperfectly, iteratively, across decades in the direction of less suffering. The pill in the cabinet has a long and strange history. Now you know it. And now you can let it go. Let the fan run. Let the room be dark. Let the brain, that extraordinary, overcomplicated, monoam-independent organ that has been following this story for the last however long, finally slow down to something approaching rest. The chemistry will still be there in the morning. You don't have to hold it tonight. Sleep well.